Namaskaram, my name is S. Ashok Kumar. Welcome to my channel. In the last part of electron transport chain, I introduced different types of enzyme complexes involved in the electron transport chain and different types of electron carriers. After introducing those introductory concepts, now I can confidently, I'm going to explain about the electron transfer in the electron transport chain. Means how the electrons are moving from one complex to another complex in electron transport chain. Now here what we are seeing, the total board from this part to this part. The total area you need to understand is the inner mitochondrial membrane. This total from here to here, this is the total inner mitochondria, because all complexes and all carriers of electron transport chain, they are embedded within the inner mitochondrial membrane. Now you can see this below this board, this is a matrix. This one above this board is a perimitochondrial space. So for time being, you remember this one is a inner mitochondrial membrane. Okay, now I'm going to start with the complex 1. The complex 1 is NADH CoQ reductase that containing FMN is a prosthetic group. Now I'm taking the case 1. The case 1 is important. Here the case 1 is a electron donor is NADH2. Electron donor is a NADH2. Why I called as a electron donor? If you take the one hydrogen atom, this hydrogen atom, if you get slip, you can get H plus plus E minus. Means one hydrogen consists of one electron. Here you will get doubt from where this NADH2 is coming. So immediately you have to tell answer to your brain like this NADH, so many NADH2s are produced in the Krebs cycle and glycolysis. That is why what is the aim of the electron transport chain? The NADH2s and FADH2 produced during the glycolysis and the Krebs cycle, they are going to convert in the form of ATP. Here I am going to start with the NADH2. So this NADH2 is an electron donor. This NADH2 giving the 2 hydrogen to the complex 1, it convert into NAD. Okay, na? It giving the 2 hydrogens to the complex 1. That 2 hydrogens it is giving to the FMN. Automatically FMN undergo reduction. It become FMNH2. You have to understand. So this after that, this FMNH2 is giving the 2 electrons to the iron sulfur protein. Here the important thing is that this iron is accepting there's a 2 iron you need to assume. This iron is now in the oxidized state, +3 state. Now this +3 or oxidized state of iron accepting the electron from FMNH2. Means it accepted. That means that ion undergo reduction, it convert into Fe+2. Thing is, you will get doubt. Sir, this iron sulfur protein accepted the electron. What about the 2 protons? Yes or no? That you have to remember. Means while the electrons are moving from FMNH to iron sulfur protein, there is a missing of 2 protons. Anyhow, electrons are transported to the iron sulfur protein of the complex 1. Okay, right? After that, now comes to the CoQ. What I said, CoQ is a lipophilic electron carrier. This CoQ will comes to the complex 1. Because these complexes cannot move, because their size is a very big size, they are embedded, they are fixed within the inner mitochondrial membrane. So CoQ will come here. It takes the 2 electrons from the iron sulfur protein. So it took the 2 electrons from the iron sulfur protein. Because CoQ does not containing iron, in order to accept the electron, it requires the protons. Na? So this CoQ accept the 2 electrons from iron and 2 protons from where, that I will tell you. You don't mix this one. So by it undergo reduction, it become a CoQH2. Okay, it undergo reduction. Then this CoQH2, that is this one reduced ubiquinone, migrating from the complex 1 to the complex 3. Once CoQ reaches to the complex 3, it CoQH2 donating the 2 electrons to the cytochrome B of the complex 3. So while it is donating, CoQ donating the 2 electrons to the cytochrome B, but here there is a missing of the 2 protons. So total how many protons missed till now? There are 4 protons. 2 protons are missing if when the electrons moving from FMNH to iron sulfur protein. 2 more protons missing when the CoQ is donating electron to the cytochrome B. Once it is giving, these are all are complex 3 itself. 2 electrons are moving from cytochrome B to iron sulfur protein. From iron sulfur protein, 2 electrons are moving from the cytochrome C1. Carefully you have to come. These 2 electrons are actually donor. Who is the donor? NADH2. Now finally 2 electrons present in the hand of cytochrome C1 of complex 3. Then cytochrome C is another electron carrier just like a CoQ. Cytochrome C will take the 2 electrons from the cytochrome C1. Then cytochrome C undergo reduction. That is why this is also called as a cytochrome C reductase. This enzyme, this complex is making the reduction of the cytochrome C. Okay. So cytochrome C, after taking the electrons, moving from complex 3 to complex 4, these 2 electrons giving to the cytochrome A. From cytochrome A, 2 electrons moving to the cytochrome A3. So finally, the 2 electrons present in the hand of cytochrome A3. Actually, whose electrons those are? Those electrons are actually from the NADH2. Means here the electron donor is a NADH2. Finally, from the cytochrome A3, those 2 electrons are accepted by the oxygen. Now, along with what 2 protons, oxygen accepting the 2 electrons undergo reduction, results the formation of water. Now, 2 conclusions will come. In aerobic cellular respiration, what is the role of oxygen? What is the role or what is the function of oxygen? Immediately you can tell. Oxygen is the final electron acceptor. Final electron acceptor in electron transport chain. That is why I am telling aerobic cellular respiration. What is the role of oxygen? Oxygen is the final electron acceptor during the electron transport chain. One conclusion. Second one, what are the end products of aerobic respiration? Carbon dioxide and water. Yes or no? Carbon dioxide, how it is formed? The carbons of the glucose released in the form of carbon dioxide. Okay, now one point. How the water become end product? Water is formed as a result of reduction of the oxygen. So water is formed because of the reduction of oxygen in the electron transport chain. Here one fast line going here, I already explained like if the electron donor is NADH2, okay, electrons are moving from complex 1 to complex 3. Who is connecting? CoQ. Complex 3 to complex 4 connected by the cytochrome C. Finally, those electrons accepted by the oxygen from the cytochrome A3 results the formation of water. Sure, Allah. Now comes to the second assumption. If electron donor is a FADH2, in Krebs cycle only FADH2 is producing. Yes or no? In the Krebs cycle, which enzyme responsible for producing FADH2? That is a succinate dehydrogenase. That is why I said succinate dehydrogenase is a common enzyme in both Krebs cycle and also electron transport chain. That important point I will tell you. If electron donor is FADH2, case 2. Case 1 is if electron donor is NADH2. Case 2, if electron donor is FADH2, 2 hydrogens given to the FMN. Now it converted into FMNH2. Then CoQ will go to the complex 2. Take the 2 hydro, means CoQ undergo reduction now. Then CoQ will move from complex to the complex 3. Then 2 hydrogen, that 2 electrons giving to the cytochrome B of the complex 3. Here 2 protons are missing. Electron moving from here to here, here to here. Then cytochrome C will come and collect the electrons from cytochrome C1. Then go and giving the electrons to the complex 4, that is the cytochrome A. From cytochrome A, electrons moving to the cytochrome A3. Finally, the 2 electrons present in the cytochrome A3. So actually, whose electrons those are? Those electrons are from the FADH2. Finally, the 2 electrons from the cytochrome 3 accepted by the oxygen results the formation of water. Second time I am asking, what is the role, what is the function of oxygen in the aerobic cellular respiration? Oxygen is the final electron acceptor in the electron transport chain. How the water is forming? Water is formed as the end product of the aerobic respiration is formed by the reduction of the oxygen. Here what is the thing? If electron transport chain is FADH2, electrons are moving from complex 2 to the complex 3. Who is connecting? CoQ. Complex 3 to complex 4. Connected by the carrier is the cytochrome C. Finally, electrons accepted by the oxygen. Means these two cycles simultaneously cannot occur. Either if electron donor is FADH2, if electron donor is NADH2, electrons moving from 1 to 3, 3 to 4 and accepted by the oxygen. Or if electron donor is FADH2, electrons are moving from complex 2 to the complex 3, complex 3 the complex 4 finally accepted by the oxygen. Means cytochrome C is always, in both cases, cytochrome C is connecting the complex 3 and complex 4. But CoQ here, it can connect complex 1 to complex 3 or complex 2 to complex 3. If electron donor is NADH2, it is connecting the complex 1 and complex 3. If electron donor is FADH2, it is connecting the complex 2 and complex 3. This is the how electrons are moving. That is why you have to take either 1, 3, 4 oxygen or 2, 3, 4 oxygen. This is the how electrons are moving in the electron transport chain. The next I am going to explain how the protons are translocating and a proton translocation in the electron transport chain. After explaining the electron transfer in the electron transport chain, now I am going to explain the same thing in different frame, that is a proton translocation in the electron transport chain and how ATP produced during the electron transport chain, that explained through chemiosmotic theory proposed by Dr. Peter Mitchell proposed this chemiosmotic theory that I will explain. Here you are seeing the first locations clarity. This one is a inner mitochondrial membrane where the electron transport chain is occurring. This inner to the inner mitochondrial membrane, this is a mitochondrial matrix. This inner mitochondrial membrane, this one is a perimitochondrial space. This border of this board is a outer mitochondrial membrane. You should remember the location. Here, for time being, I am taking the electron donor is a NADH2. You can take FADH2 also. If electron donor is NADH2, the first electrons are giving to the complex 1. To whom it is giving in the complex 1, that is FMN. So NADH2 giving the 2 hydrogens to the complex 1, that is FMN. Then FMN is converted into FMNH2. Okay, now that means FMN undergo reduction. That is a FMN converted into FMNH2. Okay, now. Then FMNH2 giving the 2 electrons to the iron sulfur protein. It is giving the 2 electrons to the iron sulfur protein of the complex 1 itself. But while it is transferring the electrons from FMN to iron sulfur protein, there is a missing of 2 protons. Where they went? Those protons are released into perimitochondrial space. Now you are getting clarity. Then after that, from iron sulfur protein, CoQ come and collect the electrons. But CoQ does not containing the iron. In order to accept the 2 electrons, it requires the 2 protons. Who? CoQ. Then CoQ taking the 2 electrons from the iron sulfur protein of the complex one, 2 protons from the matrix. Then CoQ convert into CoQH2. Clear? CoQ, then it is go and giving the 2 electrons to the complex 3. In complex 2 whom? That is the cytochrome B. Then it is giving the 2 electrons, CoQ having the 2 protons now. That 2 protons again released into the perimitochondrial space. Okay? Now, from cytochrome B, X is called as an unknown carrier. Okay? That 2 protons along from the matrix, 2 electrons from the cytochrome C, this unknown electron carrier is accepting. That is designating as a X. Then from here, the electrons are as usually giving to the iron sulfur protein followed by the cytochrome C. Here when it is giving electrons to the next carrier, the 2 protons are released into the perimitochondrial space. Finally, electrons are moving from cytochrome C1 to cytochrome C, cytochrome C to A, finally A3. Now these electrons present in the cytochrome A3, as we know, finally accepted by the oxygen. Finally accepted by the oxygen and it converted into water. This conclusion already I said what? Oxygen is the final electron acceptor in the aerobic cellular respiration. Okay, then okay. Here the very important thing what you need to remember, Now this explained through the chemiosmotic theory. While electrons are moving from one carrier to another carrier, there is a translocation of protons. Translocation of proton from the matrix to the perimitochondrial space. Okay, na? While electrons are moving from one carrier to another carrier, while electrons are moving, protons are translocating from the matrix to the space, perimitochondrial space. That results perimitochondrial space with accumulation of so many protons. Yes or no? This stay there. For example, if electron donor is NADH2, how many protons are translocated from matrix to the space? 2, 2, 2. Total 6 protons. Same thing if you take electron donor is FADH2, only there is a translocation of only 4 protons. This makes the difference between NADH2 and FADH2. If electron donor is NADH2, there is a 6 protons translocating from the matrix to the space, perimitochondrial space. If FADH2 is electron donor, 4 protons are moving from the matrix to the perimitochondrial space. Anyhow, while it is electrons are moving, protons moving from the matrix to the space. That means there is a creation of proton motive force or proton gradient. Proton gradient means what? Proton difference. Between which environment? Between the matrix and the perimitochondrial space. This is a fundamentally important. Proton gradient is a very important word in the chemiosmotic theory. In case of electron transport chain or oxidative phosphorylation, proton gradient is formed across the inner mitochondrial membrane between the matrix and the perimitochondrial space. Okay. Now these accumulated protons, these directly they cannot cross the inner mitochondrial membrane. As we know, because inner mitochondrial membrane is impermeable to the protons. That is why only channel for these protons come back is a FO particle. That is a complex 5. What I said, FO particle is a transmembrane channel for the protons. Automatically, here there is a high proton concentration. These protons are spontaneously comes down. Comes down means comes into the matrix. When these protons are spontaneously coming down through FO particle and F1 particle, automatically is what happened? This FO particle is just a transmembrane channel, but F1 particle inside there is a rotor. Means when the electrons are passing here, automatically what is happening? This F1 particle will rotate. When the F1 particle rotates, that rotational kinetic energy it will generate. Our rotational kinetic energy involved in the synthesis of ADP plus PI together forms the ADP with PI. This is we are calling as a ATP. Okay, na? Here what is the important thing is that, okay, you understood the basic concept. For example, if you take two protons are moving through this one, okay, when these two protons are moving through this one, there is a generation of approximately 8 kilocalories of energy. For example, when two protons are moving across this one, this rotational kinetic energy generation of approximately 8 kilocalories. Out of 8 kilocalories, 7.3 kilocalories used for forming the bond between ADP with PA. What is the name of this bond? Phosphoanhydride bond. So 7.3 kilocalories of energy is investing in the formation of the bond between ADP and PA, that is a phosphoanhydride. Results the synthesis of one ATP molecule. For example, what I said, if electron donor is NADH2, how many protons translocated? 6 protons. If 2 protons translocating down, moving down, how many ATPs are synthesizing? 1. Total how many protons are translocated? 6 protons. So results how many number of ATPs? 3 ATPs. If electron donor is FADH2, there is a translocation of only 4 protons. That results in synthesis of only 2 ATP. That is why, and where the difference NADH2 and FADH2 is coming. Fundamentally, the mechanism is the same. So, while electrons are moving from one carrier to another carrier, protons are translocating, results the formation of proton gradient. Those protons, while they are coming back to the matrix, the generated energy involves the synthesis of ATP. Fundamentally, the difference comes when the electron donor is NADH2, there is a translocation of 6 protons. If electron donor is FADH2, there is a translocation of 4 protons. That proton difference creating here it is a ATP difference. That is why 1 NADH2 gives a 3 ATP synthesis. 1 FADH2 involves the synthesis of 2 ATP. This is explained through a great theory, chemiosmotic theory proposed by Peter Mitchell. This is about the electron transport chain. Till now, if you understand this concept, you can like and share this video to your friends and subscribe our channel. If you have any doubt about the chapter for the NEET exam or board exams, you can comment below. Definitely, I will respond to your questions. Thank you.